Hi, thank you so much for coming today to this Oxford Martin School talk on what turns spillovers into pandemics. And to sort of set the scene, I have this beautiful slide, oh there it is, which is from a paper by Aine et al. That sort of just is one way in which people have laid out what the various intermediary steps might be. So in general, one must imagine that there is a virus circulating within a population of animals, generally. Here it's depicted as bats. Bats are very popular, obviously, pandemic hosts, but it could be an industrial chicken farm. There could be a whole range of communities in which problematic viruses might be circulating. And then at some point, there will be contact between a human and this virus, and specifically there will be infection. So the virus will be capable of spreading within that human being. So that's the kind of first phase. And it can be governed by everything from changes in human behavior, to changes in the viral genome, to changes in the landscape of human health. All these pieces might modulate that first step. The next step is that the human who is infected transmits the virus onto someone else. So these are things, I think as Ollie pointed out earlier today, we often think of those as being the same thing, but they're actually different. A virus coming into me, or another pathogen, does not necessarily translate into one going into another human. So there will be some chains of transmission that occur. Sometimes these are referred to as stuttering chains and might be short-term. And then it'll get into, again, possibly because of changes in behavior, possibly because patterns of connectivity, it'll move into the larger population, and you might have exponential growth, which is such a characteristic of these sorts of pathogens, and potentially even pandemic spread. So to talk about this today, we have four splendid experts, and I will now summon the experts to the stage to introduce themselves. And the way this is going to work is I will ask them a series of questions that we've talked about before, and then I will turn the questions over to you at some stage after we've been through a few together. So thank you very much. I didn't say who I was. I'm Jessica Metcalf. I'm at Princeton. All right. So the first question I'm going to ask is introduce yourself.
Hey, I'm Tom Peacock. I'm a fellow at the Pirbright Institute in Surrey. These days, I mostly work on influenza viruses that jump into people.
Hi, everyone. So my name is Houriyah Tegally. I'm the head of the data science unit at the Center for Epidemic Response and Innovation that's based in Stellenbosch University in South Africa. My background is in genomic epidemiology. We've done a lot of work with identifying SARS-CoV-2 variants in South Africa and in Africa during the pandemic, and now working with modeling and integrated data sets looking at climate-amplified diseases and epidemics. Thank you.
Hello, my name is Daniel Streicker. I'm a professor of viral ecology from the University of Glasgow. I'm really interested in what drives patterns of cross-species transmission, particularly from wild animals into people. A lot of my work has been on bat-transmitted zoonoses like rabies, but I'm also interested in using sort of larger scale multi-host, multi-virus approaches to understand the sources from which new infections might appear and which pathogens out there might pose the greatest risk for humans.
Hi, I'm Aine O'Toole from the University of Edinburgh. I'm a senior researcher there, and I've been working on and interested in the evolution of emerging viruses. I guess I've been at Edinburgh for a while now. I did my PhD there and worked quite closely with the phylogenetics arm of COG UK, and was involved in quite a few SARS-CoV-2 projects, including the Pango network and Pango lineages. I suppose more recently I've been working on mpox and MPXV genomics in the last number of years, and I've got kind of long-term project going on that I work on poliovirus as well, the emergence there. So.
Fantastic. All right, we're going to kick off. So the first question I'm going to ask is, what characteristics do you think are required for a successful pandemic pathogen. And successful, I mean, from the perspective of the pathogen, not from our perspective. Do you want to kick off, Thomas?
Sure. So I guess something that has an easy time jumping into humans, either because it has a large interface or infects people very easily, is a good start for the virus at least. That's, you know, if you can't infect, then you can't cause a pandemic in the first place. But I guess there's a lot of downstream things, some of that we've been discussing today, such as the ability to then transmit onwards efficiently, going into a population in which is naive to you also helps. If people already have a load of immunity, then you're not going to get very far. Anyone else want to?
I guess I could jump in just on the kind of virus characteristic side. There seem to be certain things that could make it easier for a virus to establish in humans. That might be something like a long incubation period. Maybe that allows the virus to circulate around the world more efficiently.
And you mean asymptomatic incubation, right? Incubation is asymptomatic by definition. But
yeah,
like
that's what I meant. A
yeah.
period where someone has a pathogen inside them before it's recognized that they cause clinical disease. And so that's what would allow a pathogen to move around the world using the amazing transportation networks that we have now without being detected readily.
And there's a classic result from the modeling community, which also points out that if there is transmission without symptoms, that also gives the viruses an additional chance to fly further using those transport networks.
I'm also thinking that ecologically, the pathogens would be more successful when most of the world is susceptible to it. So when most of the world is naive to it. So either a new pathogen or something that has genetic change that is able to evade previous immunity would make the pathogen most successful, as we've seen several times during the pandemic.
Absolutely. Great point.
I think one of the key differences between SARS-CoV-1, I suppose, and SARS-CoV-2 was that SARS-CoV-2 shed asymptomatically, and it made control of it much more difficult, whereas with SARS-CoV, it was not transmitting until people became symptomatic. So it was much easier to kind of actually do epi and stamp out transmission chains of that virus rather than SARS-CoV-2. So that's a key one.
Any others? One of the things that I often think about is it wants to be virulent, but not too virulent, right? Because if I guess it's the same as whether it generates immunity or it kills its host, it can prevent its ongoing transmission. So that's one of the interesting threads. Is there a place that you would flag as being the worst place for a pathogen to emerge because presumably connectivity keeps coming up?
Yeah, very dense population and contact networks, definitely. These demographic and social enhancers would be important.
Right.
From a pathogen's point of view, the worst place would be in the middle of nowhere.
For us, I thought you meant.
So what do you see as being the rate limiting step from translating spillover into epidemic and potentially even pandemic?
Sure. Yeah. So I mean, transmission, right? Efficient transmission between people, and I guess as we kind of hinted on just now, emerging in the right place, that there's the right population density to actually, because you can be the most transmissible virus, but if the person you infect doesn't see anyone for 2 weeks, then you don't have a chance there. And I think that varies a lot between different virus families. What makes good transmission? You know, if you're a retrovirus or recent mpox, then getting into the right communities maybe that are interlinked and stuff. But if you're a respiratory pathogen, it's being somewhere where you can exploit respiratory transmission and having that efficient respiratory transmission in the first place.
I feel like I've made this so that we're all speaking as if we are the pathogen, which is maybe not ideal. Houriyah?
Yeah, maybe I'm going to give a bit of a more lengthy answer, just taking as example one successful case of translation between spillover to epidemic. I'm thinking about the Oropouche outbreak in Latin America. So Oropouche is a arbovirus, so it's transmitted by insects, so biting midges, usually within the Amazon region of Latin America. And there's been several outbreaks over the last decades. but always confined to small populations within the Amazon. And then in 2024, it became a big outbreak and jumped to sustained transmission outside of the Amazon. So it was useful to try to understand why that happened. And several pieces of work, including some of our own, has shown, I think, 3 main factors that has helped create the perfect storm. One was genetic change. It was a new lineage, a new recombinant lineage of the virus that seems to be adapted for transmission. There seems to have been some enhanced ecological settings to allow for rapid transmission. So the virus found very urbanized places with denser populations, perhaps. And finally, I think these denser populations were also more connected to the rest of the country, which then helped that onward transmission to colonize new areas. So I think in answering that question, it's useful to look at what was successful versus what wasn't before.
Can you make, is Urupuchi vector transmitted?
Yeah,
Yes, it is.
yeah.
Okay. Interesting.
Biting midges,
Right.
insects.
Oh,
Yeah.
in midges. Oh,
Yeah.
wow. Okay.
Well, I think we've talked a lot about kind of ecology as a rate limiting step, but there's also, of course, pathogen evolution. And I think it's really context dependent in a way. It will depend on which pathogen we're talking about, how important the role of evolution is. And so I'll just pick on one virus that I know very well, which is rabies virus. We know rabies in humans is typically just a dead-end infection, but historically rabies has jumped between species and established permanent transmission cycles in new species, whether that's carnivores or perhaps in bats and non-human primates, maybe. But the trick with rabies is that it's already a virus that has an extremely broad host range. It can infect any mammal. Any rabies virus out there can infect any mammal. And it uses a very broadly conserved receptor. So those really aren't the barriers for that virus to establish in a new host population. So partly that's ecology, but partly it might just be some tweaking of how the virus operates within a particular species that makes it well aligned to the ecology of that species. So that's a case where not a whole lot of evolution is required, and the evolution that does happen is probably not very predictable because there might be lots of ways that you can slightly tweak how you progress within a host. On the other hand, you have a virus like influenza, where there maybe are better established barriers. So there's differences in the distribution and type of sialic acid receptors. There's differences in innate immunity across hosts. And so in that case, maybe there is a bit more predictable evolution that you would expect to happen. And maybe that's a good thing in terms of being able to anticipate the risk that any particular flu virus will have.
That's so fascinating. I earlier today used the example of rabies as a zoonosis we never worry about as being a pandemic virus, but like, is that just foolish? And I was using the fact that we've seen it infect humans so often. We know it's going nowhere.
Yeah, human-to-human transmission is
Yeah.
extremely rare. And I think part of that is probably virus biology. Part of it is human intervention. Rabies has a very strong clinical manifestation, and there are kind of physical constraints put in place for people not to bite other people when they're having clinical rabies. So I think that's sort of thing, like what you were talking about, like the severity of disease is so high, it's a universally lethal infection. So it's unlikely to establish in humans, but in wild animals, for example, that don't have that intervention in place, and so don't have any sort of intervention stopping continued like aggression of animals, like biting each other, then that's where you can have both limited chains of transmission and sustained transmission in novel species.
So are you sort of saying the evolutionary barrier is our behavior?
Well, there is partly an evolutionary behavior, an evolutionary barrier, but it seems like it varies across species. So I did some work quite a long time ago looking at the evolutionary changes that underpinned each time rabies jumped into a new bat species, and there always was some change, but it wasn't necessarily predictable across species. So it's not the same thing changing every time. So I think the human intervention is still an ecological intervention in some sense rather than an evolutionary one.
So I guess drawing on recent experience with MPXV, what I would say is it seems that when you look kind of in the past over mpox outbreaks, we would have said that they wouldn't have caught, that there's kind of fairly self-limiting, low level related to zoonosis. And right now we have 3 distinct sustained human outbreaks ongoing, 2 of which are currently circulating in Kinshasa, and 1 which, people will be aware went global. And I suppose there's quite a few factors that have changed over the last number of years that don't necessarily have anything to do with the biology of the virus itself, but have to do with, increasing populations in urban centers, kind of increasing the human-animal interface as well in areas where there's deforestation, like increasing agriculture, and kind of increasing transport between those quite remote areas where there would be zoonosis and urban centers as well. So you're kind of increasing like the opportunity for these very small, low-level zoonotic outbreaks to then come into urban centers, and providing the opportunity for them to enter into certain contact networks that may be more amenable to the virus spreading on a large scale. So in that case, it's not necessarily even to do with the virus. It's possibly just more to do with the context that the virus is existing in these days.
So would we then predict that the null idea is we're going to have more viral spillovers and outbreaks because there's more people?
In the context of MPXV, I think this is a new thing that we are seeing, kind of the opportunity and the emergence. But also with Mpox, we have an intervention. We have a vaccine that can prevent this. So it's not a novel pathogen that we have no defense against. So not everything is lost.
I guess in that particular context as well, there's an open niche for an orthopox virus in the human population. You can see that in other areas as well. I think like paralytic enteroviruses, since polio hasn't circulated so much, and like in the veterinary world, since rinderpest was eradicated, the closely related Petit Pesta ruminans has kind of emerged as a more pathogenic virus, because it was thought that spillover of rinderpest into goats before had been basically, well, if not keeping that virus from circulating, it had been keeping disease out of the population. And there's been speculation if a measles ever, well, I mean, it's not going to be eradicated now, I don't think, but if it was ever eradicated, then there's a lot of other paramyxoviruses out there that are quite nasty, like the distemper viruses and stuff that like to jump around hosts as well. So, but yeah, I guess having a niche open for the virus helps a lot as well.
So the open niche is the smallpox, the only successful eradication to date
of a human
exactly. There's rinderpest
virus.
as well. Virus.
So it's a guinea worm.
Guinea worm as well. Yes, absolutely. But the smallpox eradication opened up an immune niche for Mpox. Is the argument?
Well, I suppose gradually, because the kind of vaccination campaign would have ended at eradication in like the start of the 80s, and people have been gradually aging out of having been vaccinated, and I suppose in recent years we now have a largely immune naive population, in addition to all of the other social factors that are changing, the urbanization and everything. So yeah.
And can I ask you a hard question? You can say I don't want to answer. Is there a reason that we have a sense of what the evolutionary trajectories are for flu that we don't for rabies?
I might have to leave the rabies side of that question to the expert. I guess we have an idea of what it's going to do in humans because we have, 100 years of data, more or less. But it's much more difficult to predict what it does in other hosts. Like H5 jumping into cattle or something like that. I mean, people said that they might have had an idea that would have happened, but I don't think anyone would have taken that seriously, especially prior to 2020, before we saw it start to jump into other hosts and stuff. So I think I guess part of this, both flu and rabies are like generalist viruses in that at least certain strains can have really wide host ranges. Flu going from birds to mammals, rabies going across, the whole, all carnivores and there are other viruses like that. SARS-CoV-2 is a clear example of a generalist as well. It will infect a lot of different mammalian hosts, and it will use a wide range of receptors from different hosts as well, which I guess is another thing that makes it successful. If a virus has kind of come preloaded to be able to jump, then that helps a lot. And it probably hasn't evolved just that for humans. It's probably going to be able to infect a wide range of hosts anyway, but we don't really understand the evolutionary driver behind like having a broad host range?
I think for rabies, the problem is lack of replication. And this is much more broadly a problem in understanding the evolutionary barriers to virus establishment in a new species. It might happen just once or twice. And so you have very limited statistical power to be able to say this is like the change that needs to happen. It might have just been the change that happened that one time. So for rabies, it has jumped between species plenty of times, but often just once into a given species. So you don't have many cases of replicated host shifts from like one animal species, like kind of 3 times into a mongoose. That's like one of the best examples we've got. But most of the time, it's just a single change. And it's also often from a different genetic background too. So it might be bat virus 1 going into a raccoon and bat virus 2 going into a skunk. So a lot of things going on that make it difficult to predict between the genetic background in the reservoir host and what needs to change in the novel host.
Great. So given all the things we talked about so far, do you think there are ways we should be rethinking the way our surveillance systems work? So are there different things we should be looking for? Are there different ways we should be targeting our effort? No one wants this.
I think what's important is going to be resource allocation and resource sharing. Clearly, we can do a lot of surveillance within an outbreak that is already sustained and established, and we'll learn a lot about the transmission dynamics, but that's often very retrospective, and that can distract us from more proactively sampling new emergences in more remote locations. So I think even for a country that has a lot of resources to do its own surveillance, maybe the best course of action or with the most incremental benefit to preventing epidemics should be to share resources with some other place where we can detect and understand these outbreaks closer to emergence than when they are already established. Yeah.
Do I look like I want to talk? um, I guess I can think of 2 things. One's an easy fix. One's easier than the other one. The easier fix is that I think we often have very disjointed health surveillance systems across like human health, domestic animal health, and wildlife health. For example, if you're a ranger in a national park in South America and you notice a bunch of tapir or peccaries dying, there might not even be a surveillance system. And if there is, then it's definitely not connected to like the human health surveillance systems. So I think that's a fairly easy thing that people are starting to do and can be done. So we have some better understanding of what's going on within wild animal populations, which might either be a direct link to human populations or just sort of a sentinel system for understanding human risk. The harder suggestion that we might think about as kind of a longer term thing to be doing is perhaps like better linkages of surveillance, kind of quantitative models to understand risk, and laboratory systems to understand the risk that pathogens pose more. So I guess I need to elaborate on that. So like we can go and find viruses. We can now have okay performing models that give you an idea of the risk that those models might infect humans. But that's really where we hit a wall. And I think we need more data to have a better understanding of, of the 20% of viruses that might seem to have a high risk, which of those really deserve more attention. And so I think we need different data streams to be able to estimate that. And for that, we need to go to people like Tom and and ask for like kind of more scalable laboratory assays. So it's all fine and well to build a machine learning model based on virus genomes or something, but that's because that's something that everyone can get and it's very easy. But once you want to add in some other factor, whether it's receptor binding or innate immunity, that's when it gets really data depleted, and you would need to do that kind of in a scalable way across maybe hundreds of viruses that you might be interested in risk assessing. So I think that's a longer term goal that we might think about whether it's worth doing.
So I think when you're talking about surveillance and where to target. I guess one of the things I think has been quite interesting is that, and we've seen it in SARS-CoV-2 pandemic, we've seen it more recently in MPXV. There will always be surveillance blind spots. You know, there's heterogeneous surveillance across the globe, and some countries are very good at surveillance, and some countries are less good, or internally they may be good, but it's on a global scale we don't know. And I remember at the beginning of the SARS-CoV-2 pandemic, we were seeing sequences in the UK and elsewhere that had been taken from cases with travel history from places that did not have SARS-CoV-2 officially, or just hadn't detected it, or it wasn't sampled or sequenced or whatever. So we had entire lineages that were based on outbreaks from certain places that we didn't officially have SARS-CoV-2. And we see the same thing for MPXV as well, for Mpox. There is kind of circulation in certain parts of the world that is not officially observed, but because we have travel histories, we know that it is the case. And I think that's kind of like maybe an interesting place to try and target surveillance, is to really focus on imports, which I think obviously we already do, but transport hubs as well, places that get a lot of traffic from all over the world, and really, because you can't surveil everything, but surveilling these kind of hubs and networks where you're getting a sample of the population, I think is quite a good place.
And I think you're hinting at one of the use of genetic data to sort of pin down origins and get a sense of the spatial spread, which is something you do a lot of as well, Houriyah, has been really, I mean, I think it's, we've come to accept it, but it's quite revolutionary. I have two views on surveillance. One is that, of course, we should, eradicate poverty and everyone should have access to care, which would help. But the other is that quite often we focus on cases and we look at sequences, which is wonderful, but another lens that is available to us that we use not as often as perhaps we could is immunosurveillance. So looking at who is and isn't susceptible to particular infections. And Tom, you mentioned one of the barriers holding flu back might be the landscape of immunity. And so often the data that goes in is just numbers of cases of particular things, but there might be background information on what that landscape of immunity is, which has the benefit of being something that is, infections come and go super quick, but our immunity is always with us. And so we have this window into the past as well as a lens onto what protection might be. I work a lot on measles, and so it's pretty, it's quite powerful in that setting.
One additional point on that is metagenomics has been brought in a lot of areas for surveillance, but it is almost exclusively on the human and wildlife. There's almost nothing being done in the veterinary sector, and there's reasons for that, because people don't like to know what's circulating and not causing disease in their animals. But it's a massive gap in terms of, because there's almost certainly a lot more going on than we're aware of, but no one wants to look.
Oh, that's a brilliant point. I always try and remind myself to say emerging from the forests or the farms, but I hadn't thought about the fact that there's a strong incentive not to look. So I'm going to ask you all, what fundamental biology would you really love to have a better handle on in thinking about this, pathway from spillover to epidemic and pandemic? I feel like, Tom, you're our fundamental biology guy.
I guess one of the things that's really difficult to look at in the lab and in the field is transmission. You can look at infection, you can get your animal and you can infect it, but doing transmission models is really, really technically challenging. Even when you set them up, your SARS-CoV-2 hamster or your flu ferret or something, you do the exact same experiment with a very similar setup to someone in the lab down the road from you, and you get two completely different results as well. So there's a massive problem with like standardization of those sorts of assays as well, which you're never quite sure unless you've done it yourself if it's going to transmit in your model. There are ways of trying to take the animal out of that and try and make something that's a bit more, because I think a lot of it is to do with the actual animals themselves, but also things like airflow and things like that, which are not impossible to control, but they're hard to control. But all these models, I think, currently leave a little bit to be desired, and you have to still benchmark them against an animal transmission model. And then for lots of viruses, we don't even have animal models for them in the first place. Or if you do, it's a pathogenicity model and not a transmission model. But I think that's something that really is lacking, but I don't see any sort of way around it.
And what could you give us an example of what biology would you be learning in terms of the, like, is it this genetic sequence? What is it?
Yeah, I guess what are the properties a virus needs to actually transmit by, you know, by the airborne route, and therefore if it was in a human go pandemic, or I mean, more likely what we'd do is we'd take a airborne transmissible virus from humans and, you know, break it so it no longer transmits, and try and work out which bits were essential, which properties were essential for that virus to do that. Yeah.
Right. Brilliant.
Yeah, actually, I was going to say something similar is adaptation to transmission mode. So I'm not working in the lab, but I feel like we played catch up a lot with SARS-CoV-2 on understanding exactly what were the transmission routes, even though we all knew it was respiratory, but was it airborne or droplets or fomites? And I think that even changed throughout the pandemic. And we only realized after there were many cases that didn't match the control measures that we were trying to put in place. And yeah, I think we need a lot more research. Unfortunately, it seems it's difficult to understand that. But I wonder, I don't know, Tom, what you think, but I mean, we talked about it a bit, but about approaches like deep mutational scanning with potentially some AI. You seem skeptical, but I just wonder, like, we need to understand that better because it's also changing for one pathogen. Now imagine when we find something new, like how do we even determine that?
Yeah, I guess those sorts of things are even more difficult when it's something that hasn't got any existing data. Even if you have something that has a lot of data like flu or SARS-CoV-2 now, you might be able to make models that you could try and make predictions based on, you know, new emerging things. But if you have a completely new virus, you might not even, as you say, know the transmission route, particularly, you know, veterinary pathogens and stuff like, where it could be a GI or it could be a respiratory, and you don't know until you can kind of try and pick apart that in the lab. Yeah, really challenging, I think.
I also think transmission is a real problem. But on my take, it's more transmission within animal reservoirs. And I think one of the real challenges we have is like a new pathogen appears, we want to know what animal it came from, and we end up getting very sparse cross-sectional data. Oh, maybe we found it in this one animal, and that animal then becomes sort of the de facto reservoir that gets all the research attention, all the downstream surveillance attention. But kind of putting on your ecologist hat, we know that viruses tend to infect many more species than are actually important for maintaining them and like in a long-term transmission cycle. And I don't think we still have any sort of gold standard way of identifying what of the infected host is actually the one that's critical for maintaining transmission of that virus in the wild. And that's important to know because that's really what's governing risk for domestic animals or people.
And I guess kind of related to that as well, like drawing on mpox again, it seems that kind of the reservoir host for mpox is it's quite a promiscuous virus. It seems to infect. It's been found in quite a lot of rodents. It's been seen in obviously non-human primates as well as obviously humans. And more recently, there's been an observed outbreak driven by squirrels interacting with non-human primates. So it seems that it is infecting quite a lot of different hosts. And what you're saying, Daniel, is about the kind of trying to catch it. Observe it. When you do a sampling, you get many negatives, some positives. I think maybe that links into what you're saying about maybe we should be immune profiling, and maybe that's what we really need is like a full immune history. I guess drawing out what of the things that the reservoir has immunity to, what of those are the important pathogens for us. But that could give you a more longitudinal thing. So we should just be going out and sampling immune profiles of all of the animals.
I think Daniel, you must have something to say about this.
It would be nice if we could go out and sample all the animals. And I think that it's more and more possible to do that. There are new technologies coming online to do sort of massively scalable serological assays to kind of look at all the antibodies against all the viruses that a human or even now animals might have been exposed to. Those sorts of assays are still in their infancy in terms of making them work well in the lab and interpretable. But I think looking forward 5 years, we'll be able to do really kind of large-scale profiling of exposure histories in individual animals.
And Daniel has been leading the charge on the bat immunology landscape. So maybe linking Houriyah's question to this. So I think, one of the things that was suggested when we were thinking about landscapes of immunity, and I'm actually going to direct it at Tom. Sorry, Tom. So we have a sense of, like what the serological landscapes look like. We have a sense of what the spikes might look like. Could we figure out, could we reverse engineer what viral receptors might sneak through our immune defenses by just, probing the landscape that we have to have a better sense of what the dangers are? Is that useful, or is that just it's a needle in a haystack type situation? And I was imagining that as being a machine learning type situation, possibly.
So I guess at a very basic level, generally things that are more closely to one another cross-react more and are going to give protective immunity. But that isn't to say that it's when stuff's very, very distantly related, it's probably not going to cross-react. But it's when something's kind of in that middle distance, it's kind of hard to say, because there might be some cross-reactivity, but is it protective? I mean, the example being like seasonal coronaviruses and SARS-CoV-2, and there's a lot. I mean, I haven't kept up with that literature that well, but my understanding is that there's slightly conflicting reports about whether some of that immunity might have been protected from infection, protected from disease, completely useless, or even actively harmful. If you're a dengue, have dengue immunity or something like that. But I guess that's kind of the spot that is really relevant, but really, really hard to try and work out if it's relevant or not.
Because there's going to be a lot more biology sitting under that high level piece. Yeah, that sounds right. So, what interventions do you think might hold most promise within this landscape, or what research do you think we need as we think about deployment of interventions in the context of spillover and epidemics? Daniel.
Okay, I thought this one might get pushed to me. So it's tricky. Like, I think currently a lot of what we do is improving interventions around outbreak response. So accelerating vaccine development, getting better at rapid identification of outbreaks. Those things are going to continue to be useful and life-saving. But we have seen that in some cases, like the COVID-19 pandemic, despite those things, we can still suffer massive burdens of disease. So on the first slide that Jess put up, there was this the first category of what's going on within the animal reservoirs, and above that it said primary pandemic prevention. And so that's the idea that we might be able to block that initial spillover event or cross-species transmission event from an animal into people. So I think that's one that is incredibly challenging but promising if we can make it work. There's several different approaches that could be taken. There's lots of interest in how things like landscape restoration, reforestation, how these things might both reduce human-animal contact and improve the health of animal reservoirs. So maybe they have a lower incidence of disease or lower incidence of infection, and maybe lower amounts of shedding of a particular pathogen that we might be concerned with. The limitation there is that some of those interventions might have a positive effect on some pathogens and a negative effect on other pathogens, and we can't currently predict that. So that's where we need to understand what's going on within animal reservoirs and how any intervention that we might implement might make things better or worse. The other thing that we can do is targeting specific animal reservoirs. So for there, I'm thinking about interventions like killing rodents or killing some bat species with the intent of disease control. But that can also have kind of counterproductive backfiring impacts. Maybe it's by behavioral change in the animal that you're trying to target. Maybe it's because you're killing the wrong animals. Maybe you're killing the immune ones rather than the ones that are susceptible to infection. So I think there's promise in interventions that are both host and pathogen specific. So vaccination would be a key area there. So one of the things that I'm quite interested in is whether we can develop scalable approaches for mass vaccination of wildlife against pathogens that we're concerned about as zoonoses. So the idea there being that if you reduce the incidence of infection sufficiently in the animal reservoir, then you've essentially reduced the risk of a spillover event. And that's of course not without its challenges and problems either, but I think it is one that has potentially a lot of benefit because we would be stopping the pandemic before it even starts. I think that's about all I've got to say there.
I think we can't not say that, of course, vaccination of humans and, you know, vaccines that are the universal flu vaccine. Tom, is it coming?
Yeah, probably about the time after the universal coronavirus vaccine.
Okay,
Yeah.
good. But obviously vaccinating us to the example of smallpox and mpox is one, right, where smallpox vaccination had the unintended consequences or the unexpected consequence of protecting us against mpox, it turns out, and now here we are. All right. Any more? Do you have thoughts? Or okay, so I will now turn it over to the audience. So raise your hands, and then Moritz, you're going to wave if there's something on the iPad. That person in the back. So I think you're supposed to wait for the microphone to come to you.
Hi, great discussion. Was wondering, you didn't mention gain of function research, which is currently banned, at least in the United States. Do you suspect that this hinder our capacity to prevent future pandemics?
I think I'll probably take that one because I work on flu. Yeah, so there are lots of ways of getting around the need to do those sorts of experiments, or doing them in lower risk ways in which although you may still be doing some sort of gain of function, you're mitigating it by, for example, using an attenuated virus background, which even if someone working on the virus got infected, they wouldn't be able to be infected. I think like we're a lot further on with a lot of the technologies. There are still certain experiments that it's really hard to take any other approach than one that might be considered gain of function. I think it also, gain of function is a really nebulous term. It can be used in a way that basically describes all virological research. Growing a virus to a stock, it might get mutations, and I think there's a danger of that sort of definition being weaponized against the whole field of microbiology. As well as that, you can use the safest systems in the world that have no viruses, but you could still be describing mutations that, if they were built into a virus, would then enhance its virulence or its ability to infect humans. So there's a whole idea now coming up about like information hazards. So although the experiment that you did in the lab has zero risk, you're putting information out into the world that someone, a bad actor or a lab with poor biosecurity could then use to generate something downstream of that. So it's a really complex area, and it's one that as virologists we have to kind of be really, really conscientious about, both from what is safe, but also what information it's safe to put out into the world at the end of the day. Yeah, I don't really have much more of an answer beyond that, other than it's difficult.
Hi, thank you. So just coming back to surveillance, I think we talked a bit about more like surveillance kind of on the like viral level. On the more population level, I'm wondering what you think about kind of some like opportunities for like large language models in combination with kind of web scraping and like social media scraping, to kind of help better detect like outbreaks in maybe like communities that don't speak like they speak really local languages or maybe aren't like as well monitored by health agencies. I feel like that's you, Houriyah.
Yeah, I'm just agreeing. We should definitely use this technology to enhance our methods of digital surveillance, we call it, to really detect these outbreaks in these still localized settings before they become established. Yeah, totally agree that we should do that. And I think it's very possible with the large language models that we have now to go in that direction. I don't know if anyone else.
On the surveillance, I'll continue. I have two questions. First, one is related to surveillance. What about wastewater? The understanding is that there is a delay in the wastewater. So using hubs, as you suggested, it's sort of not going to give us a real-time information. So I wonder what your thoughts are on that. And my second question is on breakout infections. How much of the breakout infections is continuing to gear up the spillover after the spillover happens, to continue it and to continue the transmission within that cohort?
Might take the wastewater one. So I think wastewater can be a really valuable tool. I think there's often a dialogue around it being the tool, that it's like, you know, the new thing that everyone should be doing. And I'm not sure I necessarily agree with that. I think in certain cases it can be really useful. So the project that I've been working on for some time is on poliovirus surveillance. And I think poliovirus is a really excellent use case for wastewater surveillance, because there's a very clear intervention. If you detect poliovirus in wastewater, you can go and vaccinate, or, you know, depending on what the outbreak is, maybe you don't. But, you know, there are clear interventions. It's not something that you're expecting to see everywhere, you know. So you know what you're looking for. In the UK, actually, there is a national wastewater surveillance program ongoing for poliovirus, and it has, you know, in recent years detected cases, or detected the presence of poliovirus, and then interventions have been able to be made. So I think that's a really good use case. I think in general, there are other use cases. It's difficult to kind of pin down. If you look at, say, for MPXV, it could help bridge some of the surveillance gaps in certain parts of the world. However, those gaps also exist in places where there may be a reservoir species, and detection in a wastewater is not equal to a case. And the intervention is also not quite clear. You know, if you're not sure whether it is a human case, then you know, what can you do? You don't know necessarily where that wastewater has come from. And if you're talking about, say, you know, endemic pathogens that we're seeing, or seasonal things that are kind of always present, that, you know, it's interesting, but what we observe in the wastewater isn't necessarily going to be representative of what's causing disease in humans. And also the question around what the intervention, like what you detected in the wastewater, what are you actually going to do with that information, I think is still something that's not quite clear. But I think it's a very useful tool in specific cases, and I think particularly for poliovirus, I think it's invaluable. So.
Add a point to that. Another example of it where interpretation is really difficult is when it's been used for zoonotic flu viruses, so H5N1 or something, because you don't know, one, is that cow milk that people have bought from the supermarkets and put down the drain, two, cows, waste of cows, three, human infections, four, wild bird infections. The interpretation becomes a bit of a nightmare when you have multiple species feeding into the same wastewater system.
And then the question about breakout infections, I'm never quite sure what a breakout infection is.
Oh, sorry, where the information is that there is a breakout infections happening in more immunosuppressant people, or those waning of immunity from the vaccination. So is that continuing to gear up the transmission, even though the virus maybe become less transmissible over time? Do you think there is a role there? I think there is a role there. So I wondered what you think about that.
Going going to maybe get Tom to weigh in, because I think that, I think for SARS-CoV-2, for example, where you have, possibly prolonged infections where you get a lot of within host diversity generated, within an immunocompromised individual, it's not, necessarily a more fit virus in that individual, but it's what we think, a number of the SARS-CoV-2 variants came from. So it clearly is playing an important role. But
Yeah, and I guess that's the sort of place where you might find virus evolution being driven, possibly or not, depending on the virus.
Any more questions?
So most, in fact, almost all past epidemics and pandemics have spread from animals to humans rather than being a lab leak or a biological weapon, but not all of them. So it's not 100% likely to be zoonotic in the next big pandemic. And there's also a big difference between sort of wild animal spillover and industrialized agricultural spillover. So I'm wondering if the panelists can give us a very rough subjective, percentage of how likely they think the next big pandemic is to be wild animal spillover, agricultural spillover, accidental or deliberate.
Wow, Tom.
So I guess maybe there's also a blurring of the lines in some of those, like fur farming, for example. Is that agricultural or wild animals? Because they're quite often wild animals that have been brought in, especially depending on the legality of the fur farm or whatever. So I would say it's not a binary question necessarily. There can be overlaps between those. And you are completely right. I mean, again, particularly in the veterinary world, there's a number of outbreaks which have been caused by not necessarily research related, but for example, polio actually is a really great example of a lot of outbreaks of polio currently are derived from vaccines. But similarly, there are recombinant animal pathogens circulating which have parts of their genomes from supposedly live attenuated vaccines that suddenly weren't live attenuated after a while. So it is complex, and humans do have a tendency to even through our intervention sometimes make things worse. I guess, and as you say, there is at least one pandemic or epidemic that was almost certainly released through some sort of human research behavior. A freezer was involved at some point in that, although we don't know the details. So yeah, I still think, we are hyper focused on zoonosis and animal reservoirs, and we know that that if we look at human infections, zoonotic infections, they are generally at the human animal, particularly for flu, for example, the human farm animal interface. Yeah, it's really difficult to put a number on anything, right? Because until it happens, it's easy to say in hindsight, well, obviously it was going to be that, but it's really difficult to say that beforehand.
Daniel.
Yeah, I would, I guess, try to think about it in terms of what's changing into the future. So, like in the past, it probably has been mostly zoonoses. I mean, almost definitely if you look far enough back in the past before we had laboratories. So there is some, but of course there are cases that have happened from laboratory leaks and escapes. But kind of looking forward, my hunch is that laboratory safety will improve, but human encroachment into areas, human interaction with wildlife isn't going to slow down as quickly. So I think kind of the prior level is more zoonotic than lab, and in the future that should be that division should be growing rather than shrinking. So I would think zoonoses is still going to be our main problem.
And just in terms of sheer numbers of events, so it's not 100% of pandemics that have been caused by zoonosis, but it's the vast majority. And there's so much zoonosis that we're aware of. There's so much zoonosis that we don't know about. And I think when you just talk in sheer numbers, that's by far the more likely event, even with even with the status quo of lab safety today, which is all right, I guess.
Yeah, I'm sort of fascinated by the, so WHO, maybe CEPI coined the term pathogen X, and will it be, yet another coronavirus, which means we'd have a fighting chance of a vaccine, or will it be something totally nothing like anything we've ever seen before, which might be more challenging? My bias lies with everybody else's. I do think that, there's an awful lot of things out there in the woods and on the farms, and zoonosis seems the most likely, but it's very hard to put numbers. Question there?
Thank you. I wanted to ask about mpox. So as of poxviruses, they do not have tropism dependent on like receptor entry. And I was wondering, should we focus more on viruses that can infect a broad range of cells, like within an individual, and that infectivity depends on innate immune interactions? Or should we still remain focused on like a vast variety of viruses like coronaviruses whose infectivity depends more on receptors itself? Sorry, the receptors itself.
So I'll pass this along as well, but I think there's so many other factors involved in what creates or what leads to a pathogen being successful, and that cell tropism is one thing, but I think opportunity where it is, how prevalent it is, how much the human population interfaces with a reservoir. There's so many other things that can contribute. So if you were going to just draw a line and kind of say, okay, we're only focusing on this thing because of the cell tropism, I would say you'd probably be missing out on things that cause a pandemic, like coronaviruses and flus and all of this sort of thing. So there's, I think, the whole picture that goes into what you should focus on, I suppose.
Yeah, absolutely. I think it's not an either or, right? You have to do both, because both are clearly pandemic, potential pandemic pathogens. I think traditionally people have tended to focus on entry because it's amenable to a lot of assays, and it's there's been a lot of research on it, so if there's a strong foundation, it's easier to look at. But if you're not looking at how the virus interacts with the innate immune system, then you are clearly missing out on a whole other layer of tropism and the ability of these viruses and how they evolve to interact with tropism, with the innate immunity as time goes by as well. I think is incredibly important, and I think people are moving into that area a lot more over the last 5-10 years or so, which is great.
So I think I can take one last question, and then I'll ask you guys if there's anything we should have talked about that we missed. And there doesn't have to be anything, but just so you can start thinking about that. Is there a last question? Right there at the back.
Hi, my question is about surveillance, and I was wondering if we have resources to do surveillance with in all countries and for all diseases, do we have policies that are in place to protect the vulnerable countries, which probably all we're getting is information from them, and are we able to protect them from if any epidemic or pandemics come against misuse of that information? Even the question about languages for those really remote communities, are they policies in place to actually protect that kind of information, because it is valuable, but to what extent?
That's a really good point. And I think, no, we're not protecting enough data generators in this aspect, especially in more vulnerable settings. I think it's easy to say we should do surveillance everywhere and everyone should share their data. But I mean, we've seen that in South Africa when we have shared the data, we're just punished with travel bans, even though other places then become the source of the transmission. So unfortunately, I think the answer to your question is no, we don't have enough, I think, global solidarity and policies to protect these data generators, it's just easy to say, give us the data or do the surveillance. Yeah.
Great point. All right, any last thoughts? Anyone got a burning final point? Daniel has one, I can tell. No, all right, well, thank you all so much, and thank you to the panelists for